great pleasure to award the medal to Rico Cohen, whom I've known for many years. And I explained in the introduction a number of things about him. One of them is that he has done all this very beautiful work on Snapdragon development. And the other is he's a very keen amateur artist. Well, it transpires that this year's winner is also a very keen amateur artist. What's the probability of that? Young people in the audience who aspire to winning the Waddington Medal learn how to paint. So here is this year's winner, rather interestingly, painting snapdragons. So it's just a perfect connection. And I think so this year's winner, who is a wonderful scientist, but also very active in a number of things outside the lab, and I think he brings those things into his work. So what you can see here is a very colourful canvas that he's produced to show these very nice flowers. And one of his spectacular contributions to developmental biology has come through incredibly colourful, multi-spectral imaging of the developmental system on which he's focused for many years, which is the vertebrate retina, here beautifully painted with fluorescent proteins in the very same way that he paints these beautiful canvases. And the other hobbies he has outside the lab also seem to me, he brings them into the lab to understand what's going on here. So one of his important contributions has been to establish that this beautiful organized cell-type pattern actually starts kind of rather as a fishing expedition. It's a bit stochastic. You get what you fish out of the sea, and then you sort yourself out afterwards and cook yourself a beautiful supper, which is this lovely retina. But on the other hand, there are some aspects of it that have got to be more directed, much more kind of goal-oriented, axon guidance, straight for the goal. Another one of his major passions in life, ice hockey. So as you can see, if you want to be doing fantastic world-leading developmental biology, what you need to do is to do lots of exciting things outside the lab. It broadens your mind, brings you all kinds of new ideas, and you can bring them into the lab. And that way, you can spend your life on the crest of the wave, which very much is what's happened with our winner this year, who is, I'm very, very excited and happy to announce, Bill Harris.
Thank you very much. Thank you, Adeline, for the intro, and Christine for supplying those pictures, which I wouldn't have done. Thank you for the Society for this amazing honor, which is truly a highlight, a huge highlight in my career, and none too soon, because I'll be retiring in a little while. The previous winners of this award were extraordinary, and the medal winners of various other medals this time have been extraordinary. And simply judging from the incredible talks that I've heard this year, I know that there are many others as deserving, and many more so, than myself. So I am very humbled by this medal named after the great developmental biologist Konrad Waddington, whose picture you see here in two portraits. Konrad was primarily a developmental biologist, but he also made significant contributions to paleontology, genetics, embryology, leftward-leaning politics, as you'll see in a little while, and other things, such as this book, which I bought when I found out that I was going to be the award winner. So it's an art historian book. He was an art historian, and he wrote this pretty insightful book about how the scientific understanding of the world influenced the way painters work in the 20th century. In it, he argues that there's a true communication between art and science, and it isn't all just one way, not just science redefining the way that we see the world so that artists have an inkling about it and paint it that way. And he makes a terrific argument for that. But in the end, he also says that there's sometimes that art gives back something to science, and that scientists, by looking at beautiful art or amazing art, get and thinking about it, deeply, get inspired, and have ideas. And I think that's right. So Waddington was an amazing intellect, and he could have gone down many different pathways. He chose that one, developmental biology, which was good for us. But I am using that picture as a joke, but really this picture is the most famous thing that he ever did, was to create this image, to visualize the process of developmental choice. And he conjured up this provocative landscape. The cells of the early embryo are thought of as balls at a high point in a craggy landscape. And as the balls roll downhill, they enter a system of branching valleys. Each fork represents a decision point. And for the last decade or so, I've been trying to understand how cells in the vertebrate CNS make these decisions. This is what this lecture is about, is having been invited to give this lecture gives me a chance to reflect again on our results in the context of Waddington's provocative model here. And I want to share some of the work that I've done that has made me think about this model. And so this lecture is going to be about Waddington. But first, I have to outline some of our recent work on cell fate choice in the retina and point out the main conclusions that have made me think about Waddington's developmental landscape. So we work on the, is this the one? We work on the retina, this yellow thing at the back of the eye here. It's an out pocket of the anterior part of the central nervous system, and it's like the central nervous system. Here is the retina of a human, and just a cross-section through it, and you can see many different types of cells in the retina. This cell layer is the ganglion cell layer. This cell is, these are different types of cells in the inner nuclear layer, and here's the outer nuclear layer, which is the photoreceptors, the rods and cones, and here's the outer segments of those things. So the great Spanish anatomist Santiago Ramon y Cajal studied all parts of the nervous system. He studied the retina and made beautiful images. And one of the things that he noticed about the retina is that there are many different cell types in the retina. So the different layers have different cell types. For instance, we'll talk about these more later. These red ones here are bipolar cells. They connect the photoreceptors to the ganglion cells, which send the message into the brain. There are many different types of bipolar cells, as you can see from this beautiful drawing of Ramon y Cajal. So there are probably about 100 different cell types in the retina, but they come into cardinal classes, and that's all I'm going to be able to tell you about because we don't really know too much about fate choice, about the subtypes. Now, the retina starts, as I said, an outpocketing of the neural tube here, anterior neural tube, and it's a pseudostratified neuroepithelium. And now I'm going to tell you a romantic story. So Christine Holt, who's my wife, was just finishing some experiments to reveal how growth cones of retinal ganglion cells send out their axons in the visual pathway and make the first decisions. But she hadn't seen yet the growth cones as they very first emerge from the cell bodies. She was doing this technique of injecting dyes into cells at early stages, and we were driving home along the coast of California, having had a lovely seafood dinner in Del Mar or Solana Beach or somewhere, We were newly married at the time, interested in reproducing ourselves. But we were also nerds. So as the sun turned red and was beginning to set and dipped into the Pacific, Christine looked at me and murmured softly, I should say, "What if we injected the cells just before they were born?" "Well," I said excitedly, "then we'd see clones. Let's do it." So Christine and I injected, this was in a frog, and this was back down in 1988, and here we've injected a single cell, and then we let the embryo develop for a little while, and we saw clones. So sometimes single cells made clones of 3 cells, but sometimes they were these 3 cells, sometimes there were 3 photoreceptors, sometimes they were bigger clones. Could never predict what was going on. There was a lot of variability. And at the same time as we were doing this and finding that clones varied widely in size and composition, we were wondering what all this variation was about. Is it the exposure of identical progenitors to various environments as they go through their movements in the neuroepithelium? That's what we thought, because that was kind of modeled after what Drosophila was, the way the retina of Drosophila, the thinking was at the time. Or it could have been, as Martin Raff suggested to us, that all clones start out the same, but then random cell death sculpts clones of different possibilities. And Martin Raff, with Michel Caillouet, went on to study this in vitro by taking progenitor cells and putting them in tissue culture and watching the clones they made. And without any cell death, they made clones that were variable in size and composition, just like in vivo. But to answer this question, I went in a different direction, which was to the zebrafish retina, where I was hoping to see clones evolve in real time by making movies of them. And this is a second story that I hope you find a little bit humorous. Although Talak, my first PhD student whose work this is, probably wouldn't find it humorous. He's gone on to be a great guy, and he's a neuroradiologist now. He did like the work he did in the lab, but he was a medic first. So I turned to him when he was just starting in the lab, and I said to him, "What I want you to do is to follow a clone all the way from its born until it makes all the cells in the clone. Simple PhD project, just, if you can do that." And Talak made great headway at first, and he made this movie, which I will play by hitting, oops. It's hard. Here. And you can see this cell going up to divide, making 2 cells. And basically, that's as far as Talak got. He was able to see the first cell division, but that was a far cry from where we needed to get to. But he made a lot of excellent observations on that, the way that cells divide, including, you probably saw there was a little strand, a process, a string that held the cell into place, which has turned out to be kind of important. But we wanted to get to a developed zebrafish retina. This is about 72 hours. And these cells, of course, are falsely colored in with Photoshop. But you can see these are the different cardinal cell classes that we're talking about. These are the bipolar cells we mentioned before, the retinal ganglion cells that send their axon into the brain that Christine studied so much, rods and cones. These are called horizontal cells. Green ones are amacrine cells. And there's one intrinsic cell called the Muller glia. Now, recently, and thunder stolen already, Shana Almeida in the lab has been able to label all these cells with fluorescent proteins driven under the promoters that are cell type specific. So it's kind of like a rainbow. Every neuron in the retina is labeled. Glial cells aren't labeled yet, but we are now making those fish to have another color with a... So basically all the neurons in the fish are labeled different colors, and it gives you a real good sense of the architecture of the retina, the cellular architecture of the retina, that you don't get with a rainbow, which is more speckled and random. And you can make, one of the things you can do in zebrafish besides labeling cells like this, is you can make clones. And this is, if you take one of those progenitor from that fish and you put it into an unlabeled fish, you can see a clone. And you're just spinning it around, so you can see the different cell types and the different layers. So you really want to see this thing unfold in real time. Unfortunately, these markers come on as the cells differentiate and not when they're dividing. So we have to find a different way. One of the ways that people have seen clones develop in the retina is by using just an H2B GFP, a histone-linked GFP. And this is work from Brian Link's lab. And here we're going to follow maybe this one or maybe this one. You can follow either one of them. If I can find this little thing again. So yeah, so this is 4 cells now. And then they go up to this apical surface to divide, and now there's 8 cells in this thing. And they're still dividing, so they weren't finished yet. If you follow clones at this time, they'll make clones of about 20 cells. Still needed to get there. So then a very talented postdoc in my lab, Ji He, came up with this strategy, which is to use a Kayeda dye, which you can switch, photoconvert from green to red whenever you want. And first of all, you turn on the Kayeda in some cells, and then you find a nice cell in the retina that isn't surrounded by too many other cells, and you photoconvert one of the green cells red, and then you can follow that cell. And with good confocal microscopy, you can get a clone like this. This was done from a 2-cell clone, made one of them red, and so, oops, didn't work. Oh, I know what I'm doing wrong now. I'm hitting the wrong arrow, sorry. Would it just work if I did this? No. Sorry, it's okay. Next slide. Oh, yeah, sorry. I would have told you the same thing again. So, so now you can see this, and you can pretty much tell what all the different types of cells are, even though they don't have all those great labels, because luckily the retina is layered and the cells have particular morphologies. So with this kind of technique, what Ji did was he labeled clones at 24 hours, labeled cells when the animal was 24 hours post-fertilization, 72 hours post-fertilization. The retina is mostly made. And you see you get a variety of sizes of clones. The smallest ones are about 3 or 4 cells. The largest ones are almost 30 cells. And the average clone size is about 12. And there's even-numbered and odd-numbered clones. But if you make the clones at 24 hours and look at them just halfway through this period at 48 hours, now you see mostly 2-cell clones, 4-cell clones, 6-cell, 8 cells. So most of the stuff that's happening in this early phase is more symmetric. And then to get odd cells, you need to have asymmetric kinds of cell division. And here's another important thing that was done. And it's I think, important for developmental biologists studying clonal development to think about this, which, if we hadn't done it, we would have gone off on the wrong track for a while, because the first way we were following clones, the label that we were putting in there was actually killing the cells. And we were getting a variety of clone sizes, but cell death looked like, it looked like Martin Raff was right, and cell death was actually molding the variability of clones. That was our best way to describe what was happening. But when we compared the way those clones were growing to the way the retina was growing, we found those clones were growing a lot slower than the retina was growing, as if they were dying out. But with this new technique, Xi was able to show that the average size of clones, remember they're variable at all this time, is growing at the same rate that the retina is growing. Therefore, the clones are truly representative of the way the whole organ is going. And we can have confidence now that these clones are representative. And with that kind of data, how fast the cells were growing, whether there was symmetric divisions early on and asymmetric divisions later on, and when those asymmetric divisions were occurring, we developed with Ben Simons a very simple theoretical model that has a stochastic period in it. At the beginning, everything is along this pink line here, and that means one progenitor cell produces two progenitor cells. Then for a period in development, there's that drops, and there's a chance that you can make an asymmetric cell division. One progenitor makes a progenitor cell and a differentiated cell. And then there's another period, a third period. This is a simplistic model in which symmetric differentiative divisions dominate. And basically that pulls all the cells out of the cell cycle, because the chances of that happening are greater than anything else, and all the progenitors get depleted. So a simple model with a little stochastic element in it. And that predicted the results that we got when we started clones at 24 hours, looked at them at 72, when we started them at 32, looked at 72, when we started them at 48, looked at 72. It did a very good job of predicting. So from this, we inferred that probably all progenitors are equal at the beginning, and they go through a stochastic time where they might choose this thing and they might choose that thing. Oh, yeah, and he was also able to make some full movies, and these are cells where he's tracked the full lineage from 24 hours on. And you see there's variability in the way that these clones develop. But that could be due totally to the stochastic way. So here is a whole clone developing from the red cell we're interested in here, okay? Well, I don't know what happened there. I thought I hit the button. Come on, Bill. Here we go. Yeah, so now it's 2 cells, now it's 4 cells, and then it gets to the end of the 72-hour period, and it's 8 cells. It's made 2 pairs of photoreceptors and 2 pairs of horizontal cells. And if you take movies like that, and you can map out when all the cell divisions occur and whether symmetric or non-symmetric, you get the model predicts in the retina, we'll see curves like this of the PP, PD, and DD divisions, and this is what we actually observe from all those live movies. So we're pretty confident that the model isn't totally crazy. It does predicts things. But what it doesn't predict, it does a good job on predicting the size of clones, but not a good job at predicting what types of cells are in those clones. So over many years, our lab and others have started to find the transcription factors, and these transcription factors, the promoters of which were driving those fluorescent proteins in the beginning, gave the beautiful retinas, like this one here, atonal 7, which is necessary for the differentiation of ganglion cells. Comes on very early. On this one here, PTF1A, which is necessary and sufficient, both of them necessary and sufficient, for making the horizontal cells and amacrine cells. And the VSX genes are important for bipolar cells. And we don't know what is important for photoreceptors at this early stage. So using that, and also the knowledge that atonal 7, when it's expressed, forces cells into an asymmetric cell division, which we'd found out earlier, and that PTF1A forces cells into a symmetric differentiative division. So these cells not only affect fate, but they also affect division patterns. So that made us able to make a simple model, which is basically like this, that cells make a choice at some point to express atonal 7 or PTF1A, and they could have atonal 7 and no PTF1A, or they could have atonal 7 and PTF1A, and they could have PTF1A but no atonal 7, or they could have neither, you know. Those are the 4 categories that are possible. And we say, okay, to get the stochastic thing, all we have to assume is that when these things come on, and they come on and off in a fairly short window in development, that they do it independently and probabilistically. So there's a probability that atonal 7 comes on, a probability that PTF1A comes on. There's a probability that they both come on, which is just the multiplication of those 2 probabilities. And so we make basically the same model, having fit those 2 probabilities. And there's also a probability that cells will come out of the cell cycle without expressing either of them. And that's the probability of neurogenesis, which came from the previous model. We didn't have to fit very many constraints to it. I mean, it was a well-constrained model. We didn't have to fit very many variables, just 2. And to test this idea, what we did is we transferred some cells from a well-labeled embryo that had nuclei labeled and PTF1 labeled into an unlabeled animal. And when the animal was 24 hours old, this cell is just about to divide here. We picked retinas that had just 1 labeled cell in it so that we could see the clone develop. And then we got clones. And these clones basically are 1 cell cycle earlier than the clones that we started by photoconversion, and they're twice as big. And I'm not going to take you through all the modeling and stuff like that, but the theory and the model from this not only gives you the right distribution of numbers of cells in these clones, but also gives you a very good account of the different cell types in these clones. And not only the averages, those first-order statistics, but second-order statistics like the shape of the distributions of the different types of cells in the clones. So again, it's a very crude model, but it does not a bad job in giving an insight into the idea that maybe there's this stochastic indeterminacy about whether you fire this gene or whether you fire this gene, and maybe they're not talking to each other when they fire, and maybe that has to do with fate choice. So the model that I came up with, the brilliant model that I came up with to describe these results is shown here. So clone pot, zebrafish retina, you pull the handle and you get various things coming up independently, and that determines what clone you get. But actually a much better model of what's going on is this one, as I realized, Waddington's model. Because the ball can choose to go down this side or choose to go down this side. How does the ball decide? How did Waddington think the ball chose one pathway or another? And here, I'm going to read this out, so you don't have to. In such an epigenetic landscape, and by epigenetic, he didn't mean the kind of epigenetics we're normally talking about here, but how the genes pull on this landscape to make it the way it is. In such an epigenetic landscape, there are branching points at which a valley splits up into 2 or possibly more branches. What can one say about the question of whether a cell goes down one branch or another? If one can push at the right time, the cell can be diverted into one or the other alternatives in front of it. The period just before the branching point, during which a push will be effective in diverting the system into one or other path, is known in biology as the period of competence. It is no use to trying to act on the system to divert it into a particular branch until it has become competent to respond by going down the valley towards which you have pushed it. Equally, of course, it is not advisable to leave the ball pushed too late. Once the system has started to go down one branch point of valleys, if you want to divert it, you will have to push it right over the hill between them. Effective revolutionaries like Lenin have been brilliant in choosing just the right time to give a push to a society coming up to a branch point in its stability system. So you can see his left-wing leanings were straight into developmental biology. So I am suggesting that Waddington's epigenetic landscape, at least in the fish and in the mouse retina, which we've studied, is more like this. Thank you. So I'm going to end here by showing you the one thing that I would change about the Waddington model, having thought about this. And it's a simple change, which is basically adding this to the picture afterwards. You could do this experiment 100 times, and you'd get a distribution that's something like this. There wouldn't be many balls where there's a little hill, hardly any balls where there's a big hill. And if the ball was just being stochastically choosing things because they were possibly, you could, like those balls falling through those things, those pins, then you get a distribution of cell types, and it would be about the right kind of distribution of cell types. So that's kind of how I believe it works. And, Einstein said that God does not play dice. I think Darwin said that there is no God and nature does play dice. Waddington intimated that a pusher could be involved in cell determination, but Bill says there's no pusher and that cells are like dice. And these are the people I'd like to thank. First of all, it's my wife, Christine. This is our first grandchild, born a little prematurely, our daughter. Christine and I started this work. Tilak did the first cell division. Shana did the multicolored retina. Ji did those clones that you could follow from start to finish. Ben Simons helped us understand what was going on theoretically. Henrik helped with this. He was the major author on the genes that are involved in clonal cell fate determination. The Wellcome Trust supported me through all this time, and you listened to me, so thank you very much. Thank you all. I'm happy to take questions if people want to. Yes, please.
Thank you for a very entertaining lecture. So my question is whether, did you observe any differences in your data where you show there's variation in the clones that are made, whether the ones that divide asymmetrically, I mean, so what I'm getting at is, did you see a correlation between the division that might have happened and the variation that you see in the clones?
Right. So one possibility, this kind of idea that there's an explanation for the variability that you can see is a very powerful one. In fact, in some systems, there's an orientation of division that's really important, like in the Drosophila testi, and even at the very edge of the retina. And what people have found in the central nervous system is that cells divide in various orientations. And there's a big controversy about whether apical-basal division is a more differentiative division. In the zebrafish retina, that's been checked out, mostly by Karen Norden, and the orientation of division doesn't seem to matter on the clonal outcome. Whether the cell inherits the basal process or not probably doesn't matter. There are other possibilities. The centrosome, which centriole do you inherit, may be important. But I don't think any of those things. I think there's just noise in the genetic network, or that the trajectories of cells that they take, and I didn't explain all this, but the cells, when they are dividing or moving up and down through this neuroepithelium, also in a very stochastic way. And maybe they're getting exposed in a variable way to notch, for example, as they move up and down. There's some evidence for that, and that affects clone size. That's what we're going to check out next. What Afnan's going to check out. My graduate student, and sitting next to him is my other last graduate student, Megan Wave. They both have posters here. Go see those posters. Yes, Corinne.
Bill, you know, Waddington and you always speak about just one ball, but I was wondering how much your stochastic probabilities are influenced by the neighbors. So
yeah.
How much, you think, your neighbors influence the way you decide the path?
Yes.
Did you look at how much?
Very interesting question. And Afnan's PhD project is to look at a whole hunk of retina. It's a simple PhD project. He's looking at a whole hunk of retina, and he's looking at all the clones, the whole clonal history, and all the movements of all those cells. So a dense four-dimensional reconstruction from 24 to 72 hours. And in that data set, we will be able to extract interactions. There are some interactions we know, because if you transplant a cell, a wild-type cell, into a mutant that doesn't have any retinal ganglion cells, for example, there's a slight change in the probability of distributions. But it's more like the change of probability of distributions you get in a dice if you wiped out the number 6 and you rolled the dice a number of times, and you'd see higher percentages of all the others. But it isn't quite that, so there's a deviation because the clones are slightly bigger than you'd expect. So there is some kind of interaction going on, but it's a minor effect in that thing. But the question, when two cells are moving against each other and things like that, a bunch of big clones surrounding another clone, does that make that other clone smaller? Maybe some of the variability has to do with that. We'll find out. Afnan will. Yes?
Thanks for a really interesting lecture. When I walked into the room, one of the few things I knew about the retina is that it's supposed to be a bit rubbish because it's inverted. And no doubt you've had plenty of time to reflect on that. And would we be all better off if our retina were turned inside out?
I don't know. I think Simon Laughlin would argue that, yes, we would be better off if we were more like squids. We'd be able to focus the light right on our photoreceptors without interference. But Christian France has done some work that says that Muller cells, which have end feed at the edge, act like light guides a little bit and help guide the light better to the photoreceptors than it would be without those Muller glial cells, at least, and almost as good as a squid.
Almost as good as a squid.
So that really shocked me, because I thought that neural progenitor cells generally went through some sort of clock and were in different states at different times in the process.
Yes.
But at no point did you talk about time in this process. Is the stochasticity all happening at the end?
No, that is a really good, that's a really good point and a really good omission, a really bad omission from me. There is what's called histogenesis in the retina, where different cell types tend to be born at different times. And one of the early things that we found was that that's true, but there are overlaps. So while ganglion cells are being born, although they're the first to be born, at the same time, all the other cell types are being born. And if you look at clones, and sometimes the cell division produces, a photoreceptor in a bipolar cell or a photoreceptor in an amacrine cell, which tend to be born at different windows, yet within a single clone, they're both born at the same time, of course. So while there is a drift, there's a stochasticity going on at each time. You don't know what's going to happen at each time, but the probabilities are changing. And our model has probabilities changing over time a little bit. Only proliferative divisions at the end, only asymmetric divisions at the end. A brief time window when atonal 7 and PTF are on together, which define a window in development. After that, they're off, and then they can't make ganglion cells and amacrines anymore. So there's histogenesis that has to be folded into this, too. And we think that things like the temporal coordinate genes in Drosophila that were described in the central nervous system there may also be operating to some extent in the retina to help things move through phases.
Okay. And I'd like, in that case, just about, to thank Bill again for a brilliant lecture and many wonderful contributions to developmental biology, and really importantly also many wonderful contributions to the developmental biology community, including people here and well, worldwide. And as I say, just fabulous winner of the Waddington Medal, exemplary, and let's thank him one more time.
Is that okay?